And again, I'd like to thank the organizers, Olivia, Maha, for organizing this wonderful event. And it's a great pleasure to speak to this audience. And I will have to admit, it's hard to keep up after the last talk. It was really inspiring and encouraging to see that the human embryology in the next few years, in fact, the human embryology take the center stage of developmental biology in the near future. So just to continue this discussion, I can hear there's some echo. I'm not sure where it's coming from. Okay, I muted myself. And so it's great to see that the human embryology will take the center stage of developmental biology. That's my view. And hopefully, I guess you will see some contributions from engineers like us using stem cells, human pluripotent stem cells, in conjunction with bioengineering tools to build useful and faithful models to further advance this field. All right, so there's a good reason, in fact, why we're very interested in using stem cells to build models to advance human developmental biology. And I guess without further, I don't have to go into details why studying human development is important. But nonetheless, I should point out, I think after the last talk, it's very, very difficult. I should point out it's very, very difficult to have access to early human embryonic tissues, particularly, I guess, from implantation up to the first, I would say, till, 4 weeks after conception. And afterwards, you might have the access to human embryonic tissues become more accessible, more available. But nonetheless, the first few weeks after implantation, the human development is critical and essential. You can see panel A here. This is the implanting human blastocyst, pre-implantation blastocyst attaching itself to the maternal uterus, setting up the stage for post-implantation human development. You can see from the scale bar here, it's rather small, about 100 micron in diameter, containing about 250 cells. And you can imagine that after implantation, basically they will disappear and invisible for observation. As discussed in the last talk, and the first few weeks after implantation is critical. First, we know that, for example, and the clinical data suggesting that the first few weeks after implantation and even before implantation, and there's significant chance for pregnancy failure. That's this famous, what we call iceberg of successful pregnancy. And also we know that the first few weeks during the pregnancy, the basic body plan will be established and all the organ rudiments will start to develop. And there are major risks of birth defects that will happen within the first few weeks of pregnancy. So we have very difficult access to human embryonic tissues, not to mention that to study human development. But given the importance, we ask whether we can leverage the early developmental potential of human pluripotent stem cells as the cellular tool, and in conjunction with bioengineering tools, can we build faithful models in a dish? And can these models serve as useful experimental and discovery tools to advance human developmental biology? And furthermore, can we use such models to open a path to generate functional cells, tissues, or even organs in a culture dish for downstream translational applications? So our vision for this direction is to integrate bioengineering tools and stem cell biology and developmental biology to build faithful and useful models. And as you all know, in fact, in natural development, we have single fertilized egg, and through natural development, the cells will continuously divide, differentiate, and into different lineages, and they will start to somehow organize, develop functional tissues and organs. And this intricate, self-organizing properties and the tissue-tissue interactions and intrinsic, developmental potential contained in the cells. And in our approach, and we use human stem cells, and in particular, human pluripotent stem cells, and including both embryonic stem cells and induced pluripotent stem cells as building blocks. I should point out in our research, we exclusively use what we call human pluripotent stem cells as primed pluripotency state, and which means that developmentally speaking, they are very similar in terms of properties, very, very similar to perigastrulation pluripotent epiblast cells. That's where the cell types we use, the cell types we use, and the perigastrulation become where we are really interested to model using our models. And of course, our approach will be really, based on developmental and stem cell biology, and again, in conjunction with engineering approaches and tools, and to build models both embryo and organ models. And of course, in our view, and there are a lot of tools that we can incorporate into this research. For example, there are biomechanics tools we can use to control the tissue morphology and apply mechanical forces, understand how biomechanics play a role in tissue development. So there are many, many different tools, and we can also further utilize, for example, microfluidics. I will show you some examples to superimpose defined controllable morphogen gradients to control tissue development. And also, we are very interested in incorporating theoretical and computational methods, and either theoretical or agent-based or even AI-based tools into our research and to advance this field. And there are also advanced bioengineering techniques and 4D imaging, live imaging, and genetic barcoding, genetic tools. And then many, many tools now become available, and we can incorporate such methodologies and with our models to really advance fundamental understanding of human development. Hopefully, I will illustrate, be able to convince you that there are good opportunities there, and the field is really ripe for further, development. So in our research, we're very interested in a few different, milestones, phases of human development: gastrulation, brain development, heart development, and gut tube formation. And these are a few different directions we're pursuing in the lab. And we're very interested in using such models as experimental tools, discovery tools, to understand, for example, the molecular and genetic mechanisms behind the cell fate decisions, tissue patterning, and the tissue-tissue interactions. And because we are engineers, I should point out, I'm in a mechanical engineering department, and mechanics is always something in our mind, biomechanics, forces. We're very, very interested in using the models we have been building to understand biomechanical principles underlying, for example, different morphogenetic events, including, for example, tissue boundary formation, cavity formation, and tissue bending. These are biomechanical principles underlying, early development. So just to highlight, to give you an overview about where what type of, the different projects ongoing in the lab. I grouped different projects into two different camps: the paragasulation development and early organogenesis. So paragasulation development of almost a decade ago, and somehow, through some unexpected discoveries, and we realized that human pluripotent stem cells can surprisingly spontaneously give rise to what we call amniotic sac. And don't worry about the terminology, I will explain more. And somehow during this paragasulation development and this amniotic sac formation, there's also a very important cell type, primordial germ cell. These are the precursor cells for sex cells, germline, in the human body. They will emerge in the tissue, and we're very interested to study the origin and genetic regulation of this very important cell lineage, cell type. And there's also the yolk sac is also been developed during paragasulation. And one of the functional feature of the yolk sac is blood development. In fact, blood cells will first develop in the yolk sac in mammalian development. And we're also very interested in using our model, and we have a yolk sac model to understand how blood cells, blood lineages been developed in the yolk sac.
The germ cells, is that from naive or from primed?
So all the models we have developed and the starting cell population are from primed human pluripotent stem cells. So the early organogenesis, as I mentioned, I will highlight the nerve system development models that we have been, pursuing over the last 5-10 years. And I don't think I will have time to cover heart development, gut tube development, and somite formation. But nonetheless, I really like to just discuss a few selected examples and to highlight where we are and what type of research activities and questions we can pursue that we are excited about. So the first part of my talk, I will focus on modeling paragasulation human development. All right. So this is a little bit more, just to give you a little bit more information about what's happening in the human embryo soon after its implantation. And this is the human blastocyst. And you can see that the pluripotent epiblast cells are surrounded by some extraembryonic tissues. You have the hypoblast that will give rise to the yolk sac. Then trophectoderm will give rise to the placenta, contribute to the placenta development. Soon after implantation, you can see from the cartoon that the epiblast compartment will self-organize, open a cavity, and somehow with the cells next to the invading trophectoderm, they spontaneously differentiate. and break symmetry, forming this what we call bipolar amniotic sac tissue, with the remaining cells remain pluripotent, but soon after they will undergo gastrulation and differentiate into different germ layers to organize the cell body, to establish the human body plan. So this is the corresponding sectional images, and again, there's some available kinetic stages, sectional histology, I guess, human tissue samples, and you can see this is amorphous epiblast cells, and soon after, you can see they form a very interesting bipolar structure with the columnar epiblast cells. Then next to it, the squamous, the cells become squamous. These are the amnion cells, amniotic cells. And the amnion cells, of course, eventually they will develop, further contribute to the amnion formation, amnion membrane formation. All right. So about 1 decade ago, I had this very talented PhD student, somehow in the experiments, and without providing, because this has been published, available, and somehow in the three-dimensional culture system, and we were really just seeding human pluripotent stem cells randomly in a 3D culture, even without adding any soluble inductive signals, exogenous soluble factors. And somehow you can see from the videos here, the cells, they exhibit amazing self-organizing properties. And you can see from the video, I will explain it. So they form a small cavity, and notably, and soon after, you can see some cells as one pole of the cell cluster, they become squamous and flattened, and the symmetry breaking and forming this bipolar structure. And even within a subset of such structures, we start to see additional phenotypes suggesting that the structure, the bipolar structure, they can even undergo progressive development. So you might notice that they break symmetry. Soon after, you can see cells delaminate from the columnar pole. The cells, they start to undergo what we call gastrulation-like events, just like what they're supposed to do in vivo, even without adding any soluble, exogenous soluble factors. This really triggered the imagination, I guess, the belief that the human pluripotent stem cells, maybe we shouldn't be surprised. And we have to trust the cells, in fact. And they really have this early developmental potential and self-organizing properties. Somehow,
what's the base media you're using?
Either essential 6 or M-teaser. And these are basically the basal medium that people use to culture, maintain human pluripotent stem cells in a culture dish. But of course, we are 3D culture in the sense that the cells are embedded in gel, like we're using matrix gel, and they're indeed embedded in diluted matrix gel. And I have to admit that the matrix gel itself does contain, I guess, unspecified factors, and we believe that likely those unspecified factors, including, say, BMP signals, BMP molecules, might play a very important role to drive symmetry breaking, for example, because we do have data suggesting that BMP signaling is required. BMP activity is required for symmetry breaking in this system. So of course, following this early discovery, and we decided, because this is a conventional 3D culture and with very limited controllability, as I mentioned, that only a subset of cell clusters, less than basically 10%, will undergo this intricate, very interesting, like a self-organizing tissue morphogenetic events. So I have this very talented postdoc. We decided to bring in additional, I guess, bioengineering tools to make the model much, much more robust, controllable, and scalable. So in this sense, and we developed a microfluidic system, and without providing technical details, and this microfluidic device allows us to position cell clusters in prescribed locations within the device. And this microfluidic system allows us to superimpose external morphogen signal, soluble factors, to break symmetry, to control the symmetry breaking events. And it becomes very, very effective. You can see it from the videos here, and it's going to replay and capture synchronized development of 5 different cell clusters. And you can see cavity formation. And if you watch very, very carefully, and we are adding BMP4 into one of the channels, so therefore only the cells directly exposed to BMP4 will differentiate into amniotic cells. And so we can control the symmetry breaking events. And soon after, you can see after symmetry breaking, only the cells as this pole, they start to delaminate. And again, they're undergoing what we consider as gastrulating-like events. So following this work, and so now I'm going to talk about unpublished data. But it turns out early post-implantation development, the amniotic sac formation, and during amniotic sac formation, there are some other cell types that will co-emerge in human development, So let's take a look of the zoom in view, and this is CS4. This is basically pre-implantation human embryo blastocyst. So this is the early post-implantation. And you can see that this is the epiblast surrounded by hypoblast and trophectoderm. Again, the hypoblast will give rise to the yolk sac, and trophectoderm will give rise, again, contribute to placentation. Soon after, within basically a few days, and you can see the break symmetry, you have the columnar epiblast, amniotic ectoderm. This is what we discuss as the amniotic sac. But there's also another lineage that will co-develop, emerge. This is what we call extraembryonic mesoderm. This extraembryonic mesoderm will separate the amniotic sac from the trophectoderm lineage. And now the question becomes, what are the interactions among different cell lineages, different cell types, how they co-develop, and how their interactions contribute to the robustness of symmetry breaking and tissue patterning in the context of early human development. So to approach this question, to approach this opportunity, it turns out indeed recently, in the stem cell biology field, people have developed certain protocols to obtain extraembryonic mesoderm-like cells from human pluripotent stem cells. So now we can really combine both cell types to study cell-cell interactions, whether the presence of the extraembryonic mesoderm-like cells will be able to dictate the symmetry breaking events. So to test this hypothesis, so we have the microfluidic device. We just load extraembryonic mesoderm cells into one channel, then load the human pluripotent stem cells into the other channel, and allowing the cells to self-organize. We can study how the cells will behave in the presence of the extraembryonic lineage. So now, and in this case, we're using a reporter line, human pluripotent stem cell. In this case, the human pluripotent stem cells, this is a reporter line, ILC1 TD tomato reporter line. It turns out ILC1 is a marker, lineage marker we use to tag the differentiating amniotic cells. And at the same time, these human pluripotent stem cells express EGFP, allow us to basically make sure this is the human pluripotent stem cells instead of extraembryonic mesoderms. So you can see the video here. You can see that indeed after cavity formation, only the cells most adjacent to the extraembryonic mesoderm cells, they start to show. Now you can see that. Now you can see the expression of ILC1, this symmetry breaking, and the amnion cells start to differentiate only for the cells most adjacent to the extraembryonic lineage. So that really suggesting that likely in vivo, the co-development of the extraembryonic mesoderm will ensure the robustness of this amniotic sac tissue symmetry breaking and patterning. Please.
That in the movie?
This one?
Yeah.
Likely, I think it's from the defect of the imaging. There might be some air bubbles trapped underneath the cover slip. It's not perfect, yeah, but I think it's clear enough. Yeah, okay. And hopefully at some point we will be able to deliver as beautiful as possible the videos and the images. Yeah, okay, all right. Feel free to interrupt and raise your hand. Yeah, I think this is really the good thing about this workshop is it's an open conversation, and we don't know everything we're studying. I have to admit, and early development, particularly we're talking about perigastrulation development, remain mysterious. And even when I say that, we believe that we're using what we call extraembryonic-like cells, and trust me, it's a big question mark. It's a very big question mark, and we have very limited. I guess remain to be validated. Many of the stories or I guess information we're discussing here remain to be validated. All right. So now the question becomes whether can we utilize our model to really conduct mechanistic studies. This is really, I think, a very, very important discussion. I was talking to Olivia about this particular field, and this field is very interesting and exciting. Now people realize that using human pluripotent stem cells, you can build models. I'm sure you have heard of the term synthetic embryo, and it's very eye-catching. But I think for the long-term growth and healthy progressive development of the field, and we have to use the model to demonstrate new biology or interesting unknown human biology. And this is really what we want to put the emphasis and focus on that. So we decided to focus on one interesting cell type, a human primordial germ cell, and using our model to study this cell type in our model. As I mentioned, that these are the precursor cells of functional sex cells. And therefore, it's tremendously interesting and important for human reproduction or even human evolution. And so it also turns out it has very interesting biology and also shows significant species difference among different species. For example, we know that, for example, in mammalian systems, pig or otherwise mice, the primordial germ cell marked by SOC17 and BLIMP1, the double positive cells, they will emerge during gastrulation together with the gastrulating cell types during gastrulation. But there's some limited data generated from primate monkey systems now suggesting that in primate development, this primordial germ cells, SOC17, TFAP2C, marked by the arrowhead here, they will emerge in the amniotic ectoderm compartment, in the amnion compartment before gastrulation. And also from extensive in vitro studies, we already know that. And from different species, the primordial germ cell, their transcriptional regulation programs are very, very different, very, very different. For example, SOC17 is basically required for primate human primordial germ cell development. But SOC17 is dispensable. It's not even required. It's dispensable for mouse primordial germ cell development. That really suggesting that we need to have a primate-specific system to really understand, to study this interesting, important cell type. So to approach this opportunity, of course, we first examine whether we have primordial germ cell in our system or not. It turns out we do have this cells. We do have such cell types, and you can see marked by the arrowhead double positive for SOC17 and TFAP2A. They will emerge first in the amnion compartment, and over time, and we see some interesting dynamics, they start to accumulate towards the junction between the amnion tissue and the epiblast, marked by the arrowhead. Then soon after, over time, they start to intermix with the gastrulating cells. They start to delaminate together. So there's some very interesting dynamics.
What you seem to imply is that they derive from the amnion precursors. Is that? I thought this was a bit controversial.
It's controversial. We don't know. And what we know, given the data, unfortunately, I didn't include the data. There's no concrete data to suggest where they come from. What are the precursor cells of primordial germ cells? But what we do know, I think, given all the data and including some data generated from my lab, there's a precursor population, and that can give rise to, that's competent for both for primordial germ cell like cell of primordial germ cells and the gastrulating cells and the amnion cells. And these precursor cells, their competence to give rise to all of these three different lineages, in fact, it can last for a prolonged time. It's not a short window for the competence. And we have data to suggest that. But where they come from, the precursor cells give rise to primordial germ cells, we don't know. We just know that there are common precursors. Does that answer your question? Yeah. You have.
And this is in the IPC. There is no data on this.
There's no primate data. As you can imagine, that to conduct experiment using primate system is extremely difficult. And what I was saying, that the available data generated from in vitro systems, also including in vivo transcriptome data, and people can do lineage, like mapping the lineage trajectory based on transcriptome data. That's basically the hypothesis. The data suggesting that there's common precursor population. How the cells decide to differentiate into different lineages, we don't know. But I will show you some data. We're starting to examine this important question.
So they're not from the yolk sac. I thought they were coming from the yolk sac, but I'm wrong. No?
Yeah, they are coming from I would say, the amniotic sac, but eventually over time, they migrate towards the yolk sac.
Okay.
That's a later stage.
Okay.
They migrate towards the yolk sac. Yeah. Then eventually they migrate along the hindgut.
Yeah, okay.
That's a later stage. But early stage, primordial germ cell, they're happening in the embryonic compartment. Yeah.
Because in mouse, they do not derive from the same precursors. I mean,
no, it also turns out in mouse, for example, and for sure in mouse, and the primordial germ cells coming from the gastrulating epiblast compartment, they co-localize in this compartment. And it also turns out amnion will only amnion will only differentiate after gastrulation in mouse. That's also a very interesting observation. And that's, again, suggesting that there's species difference in terms of the origin of the
to do with how gastrulation achieved in different species?
Very possibly true, because as you know, and the mouse paragastrulation and the human primate paragastrulation is very, very different. In fact, if you go back to this very simple schematic here, and we also know, if, for example, the actual embryonic mesoderm, amnion, epiblast, such tissue organization, they don't exist in mouse. The paragastrulation mouse structure is what we call X cylinder, and the epiblast is next to what we call actual embryonic. I'm not familiar with I guess I'm more familiar with human than mouse, unfortunately. And they have very different tissue organization and topology, and they even contain different, I guess, the actual embryonic tissue next to the epiblast is different. It's different. Yeah. Okay. So to tackle this opportunity, we decided to do a live imaging, and because indeed there are some unique markers, lineage markers, allow us to tag the development to monitor the emergence of primordial germ cells. So in this case, we're using SOC17, and we develop a TD tomato line, and you can see now this is the live imaging. The emergence, you can see the TD SOC17 positive cells, they start to emerge. But more importantly, this allows us to check the emergence of the cell types, and after their emergence, where they go. So indeed, by monitoring, by checking the migration of individual cells, and you can see that the red arrowhead, the green arrowhead mark the primordial germ cell-like cells developed in the amnion compartment. And you can see after their emergence, somehow they show some type of directional translocation. I'm not even using the word migration. So they will just kind of migrate towards the other side. And so this is I guess, the quantitative data showing the movement direction of the primordial germ cell-like cells emerge in the amnion ectoderm compartment, and versus those coming out directly from the gastrulating compartment. And they show this migration trajectories, kind of consistent with what we believe that indeed there might be primordial germ cells, they will be specified in the amnion compartment, and then later on they will migrate. And join together with the other cells. But of course, this is not sufficient enough. So what we decide to do is to use conventional but powerful developmental biology approaches to check lineage development, and using this genetic tracing approach. And in this case, we develop a Cree-responsive, I guess, Cree-LoxP system, and this ZAS-green, I guess, expression responsive to Cree expression. And this is, I guess, knocked into the ZAS-green, Cree-responsive ZAS-green knocked into the AVS locus. The Cree expression will be driven by GATA3. The reason we chose GATA3 as a lineage marker in this case is GATA3, it turns out, is a marker that allows us to check amnion differentiation. So when amnion start to differentiate, it will upregulate GATA3. So that we know that all the cells differentiating likely from the amnion compartment, they will upregulate the green fluorescent signal. So indeed, when we look at our sample at different time points, you can see that this is the early time point, this is the later time point. At the early time point, all the TFAP2C is a very unique marker, allow us to mark primordial germ cell-like cells in our system, and they are all ZAS-green positive. But at a later time point, we start to see two populations of TFAP2C positive cells. And one positive cells will be ZAS-green positive, while the other one will be ZAS-green negative. So that really suggesting that indeed we start to see two populations of primordial germ cells as later time points. And this gives us some confidence about using this Cree line, allow us to mark two populations of primordial germ cells. And of course, we can leverage this Cree line in conjunction with genetic tools. There are amazing genetic tools now available given the last 1 decade development and available now, and even allow us to do genetic screens to test, examine all the human transcription factors, their function in primordial germ cell development. So to do so, in fact, again, we have this GATA3 Cree line overexpressing Cas9 And there's this lentiviral human transcription factor library and guide RNA expressing different guide RNA to target different human-specific human transcription factors, more than 2,000. And after infection, and we can sort out the cells and then use the cells to generate our model. And of course, then, because the human primordial germ cell-like cells, they have unique markers. And or otherwise, we can use the GATA3 reporter to sort out the fluorescent, the GFP positive cells that really allow us to separate all the cell populations into different compartments, say the amnion PGCs and all the somatic cells, Then we can use the NGS sequencing and to sequence the guide RNA library to allow us to examine what are the cells being enriched or what are the guide RNA being enriched in the different cell populations that really allow us to generate, to generate a table of potential targets that might play a role in the specification of human primordial germ cells. So in this case, these are the genes enriched in somatic cells that basically suggesting that when this genes being targeted, being targeted, that might block human primordial germ cell development. And this is not surprising. In fact, the target we identified, SOC17, TFAB2C, IL-1, GATA2, and I was even saying that TFAB2C, SOC17, these are good markers. So this is not surprising because, in fact, this is almost a sanity check. And this is almost like give us more confidence. This genetic screening approach is working. And this is not surprising either because these genes are known to be important for primordial germ cell development, known to be important. So
do you think they have the same potential, these two populations? How can you know that?
This is a great question. I guess Olivia's question here, let's assume that indeed that our observation is valid. We have two different compartments, two different cell populations. In terms of function, are they different or not? And the short answer is, given the data we have, and we have done limited experiments, and as the transcriptome level, it's very hard to tell the difference. And as the functional level, well, we have done very limited functional studies. And as the current stage, based on our co-culture studies, and we don't see significant difference. But of course, we have not done a lot of long-term assays. And unfortunately, those long-term assays are extremely difficult. And in the whole world, there are only a few labs, handful of labs can do those long-term, prolong the maturation, get to the maturation stage. It's very difficult. But as it is for the assays we have tested, they appear to be similar. Transcription level and proliferation, very, very similar. Yeah. But nonetheless, so going back to the data here, indeed, we identify a few different transcription factors. And when we knock out, indeed, you can see that essentially blocked the primordial germ cell induction in our model. So this is a sanity check. But most importantly, in fact, is what are the surprising findings from our genetic screening? That's what I'm going to present. So it turns out, indeed, we also see we have another list of targets, genes, that basically is depleted in the amnion primordial germ cell population that really suggesting that these are basically what we consider as negative regulators, restrictors. So suggesting that these genes, when you block them, when you perturb them, that somehow will encourage, promote human primordial germ cell development. So because this is unpublished data, and given so my postdoc asked me, don't never ever name the gene. It's just called gene 1, okay? Gene 1, sorry, gene 1. But it also turns out this is primate specific, and which really make this even more exciting because this is a primate specific transcription factor. And so we identified, in fact, so for this particular gene, and when we knock out this gene, and this is quantitative data, we do see a significant increase of the primordial germ cell number in our system. And of course, I should also point out, we tested a few genes, and the gene number 2 here also shows similar phenotype. So again, this is very, very, to us, is exciting. And now we even identify like primate-specific transcription factors. And of course, this is single-cell sequencing data. And also you can quickly see that this is the knockout phenotype, and this is the wild type. You can see drastic difference, the population, the composition, or the percentage of primordial germ cell-like cell compare these two populations. But more importantly, now, in fact, we can further go back to the primate system now to validate the findings. And this is, of course, we cannot do that in our lab. So we collaborated, and the wonderful collaborators in China, and Dr. Tianqing Li and Dr. Chenting Zhang, and they have established in vitro culture systems for primate monkey embryos in their lab, and they can prolong the culture of such monkey embryos towards perigastulation stage. So this is the wild type, and this is the knockout for that particular gene. And you can see the arrowhead marked the primordial germ cells, NANOC, TFAP2C, SOC17, the triple positive cells. And this is the knockout. And clearly you can see this is the basic immunostaining data. And we see a lot more primordial germ cells in the knockout monkey embryo. And this is the quantitative data. As I mentioned, it turns out this particular gene is not necessarily just function in the primordial germ cells from the amnion compartment. And unfortunately, it's not. But it has drastic effect on the primordial germ cells from both compartments, the amnion compartment and the epiblast gastrulation compartment. And you see drastic increase for both cell populations, So what we believe that, given the experimental data, we believe that this gene 1 is basically a restrictor to make sure that you don't get, during primate development, there's no excessive primordial germ cell formation in early development. You just need to have enough cells, but not excessive number of cells.
You knock down or knock out that gene.
Knock out what?
Knock out or knock down that gene 1.
Oh, this is the knockout phenotype. When we knock out this gene 1, we see drastic increase of the primordial germ cell numbers in the monkey embryo. That really suggesting that this gene 1 serving as a restrictor to basically tell all the cells, prepotent epiblast, to become soma and also the amnion tissue. So it's a restrictor. And of course, now this really opened the door to understand the primordial germ cell biology, because this is a primate specific transcription factor, how it intact and how it play a role in the overall transcriptional regulation of primordial germ cell development. And
my total ignorance, I should know this, but how do knockouts in monkey embryos?
you basically, as the zygote, and then you inject, yes. Yeah, it's and unfortunately, given, I think that you almost, to study primate biology, you almost have to, unfortunately, at this point, to collaborate with folks in China or maybe Japan. And I think we are really behind. And I think we are really behind to study human biology, primate development. But nonetheless, yeah. Okay. So the rest of my talk, I'm not sure what's the timing. I didn't really watch my clock. How much time I have to speak? Maybe another 5-10 minutes? Okay. Oh, another 15 minutes? Okay, then. Okay. Then, of course, and in the field, this is a slide, I guess in the field of embryo modeling, and there's a tremendous interest from many others to generate complex human embryo models that hopefully can undergo gastrulation, maybe even lead to early organogenesis in such a way that organ rudiments from different germ layers can be generated from the embryo models. But nonetheless, we're not there yet. And there are some folks in the field, they are very interested in that direction. So they're adding more complexity, adding more cell types, and make more complex models. But nonetheless, and again, as I mentioned, we are not there yet. And I think that would be scary and might be a disaster for the field, to be frank. If someone reports saying, using human pluripotent stem cells, I generate a human embryo-like structure that can progressively develop from blastocyst to somewhere early organogenesis, I think it will be disaster for scientists in the United States. In my view, that's my view. But nonetheless, we're not, but we still have certain strategies, I think, to bypass gastrulation, to achieve early organogenesis models, and without involving intact, complicated, complete human embryo models. So let's take a look at the schematic, very, very simple schematic. And the gastrular organogenesis, we understand that after granulation, you have the 3 different germ layers, and each germ layer will be responsible for certain major organ formation. And through classic developmental biology studies, we all understand that the organ formation from each germ layer, they exhibit very significant autonomy, self-organization, and modularity. And again, I always use this very simple sentence, almost so that to show our naiveness as an engineer. So from the engineer's naive perspective, organogenesis, you have this very simple script. You have a naive cells, precursor cells, and they undergo some morphogenesis, and they start to form a 3-dimensional tissue. And of course, such morphogenesis can be driven intrinsically by genetic programs or otherwise adjacent tissues. But nonetheless, and maybe afterwards, they will start to perceive, or maybe even during the formation of the organ rudiment, they start to perceive inductive signals from adjacent tissues. So tissue-tissue interaction become very, very important. And this is what we call induction patterning. So the tissues will be patterned and different regions will become different cell types. And of course, constricted by the gene regulatory network. You can apply this script to central nervous system or otherwise the endoderm organs. And just focus on the central nervous system because that's what I'm going to talk about next. A sheet of naive ectodermal cells, ectoderm cells, and undergo neuralization, they form a neural tube. And then the neural tube will perceive inductive signals from adjacent or nearby signaling centers, say notochord or epidermis, or otherwise they are secondary signaling centers as well. And they become patterned divided into forebrain, midbrain, hindbrain, and spinal cord. And again, they under the gene regulatory network constriction and different cells in different regions, and they will differentiate into different cell types. And eventually, hopefully, of course, in development, there's also long-range interactions between different domains. And so this is what I'm going to talk about just to highlight how we can use engineering tools to build useful models. And I guess in this case, a human neural tube model. And I will showcase this to illustrate our approach. So this is another, I guess, the very simplified view of the early human neural tube development. And you have this neural tube. And somehow, I guess, during its formation, it perceives inductive signals from adjacent tissues. You can see notochord, epidermis. And somehow, people know that, again, through classic developmental biology studies, there are important soluble factors, FGFA, Wnt, rationic acid. They serve as morphogen signals to pattern the neural tube into forebrain, midbrain, hindbrain, and spinal cord. At the same time, you have the epidermis and notochord. They will secrete, I guess, anti-parallel morphogen gradients perpendicular to the neural tube. And these are important for dorsal ventral patterning of the neural tube. And of course, there might be many other soluble factors, but these factors are known to be important. So to achieve this, I guess to demonstrate, we can establish a patterned human neural tube model, and we develop another microfluidic system, and this is a former postdoc, as well as graduate students. They did PhD first and then stayed in the lab as a postdoc. That's what I find as a winning strategy is to keep your best graduate students as a postdoc in the lab. And once in a while, sometimes it works, and most of the time they will go somewhere else. They will come to Boston. Yeah, that's what happens all the time. But nonetheless, so we use a microfluidic system, and this device allows us to pattern. You can see that we can pattern a human pluripotent stem cell colony in the center of the channel. And you can see this channel contains 3 different channels. And these 3 different channels, each one of them have reservoirs connecting to the ends of the channels. So now we can add soluble factors into each reservoir to establish morphogen gradients, either along the length of the tube or otherwise perpendicular to the length of the tube. In this case, we're adding Wnt, FGFA, rationic acid only into one of the reservoirs connecting the center channel. And of course, there's a basal background, medium background. We have DuSMAT inhibitors. And then basically, you can imagine that through passive diffusion, they will form morphogen gradients along the length of the tube. And this is for rostral-caudal patterning. And for dorsal ventral patterning, we're adding BMP4 and rationic acid and sonic hedgehog into the other channels. And you can see that will allow us to establish anti-parallel morphogen gradients of the signals perpendicular to the length of the tube. So the videos here the videos here, the top video here capture synchronized development of a few different cell colonies and during the first few days of development. I should mention that because this is a genetic approach, we can pattern numerous tissues in the center channel, or otherwise we can pattern just one tissue. Again, for the purpose of different applications, different questions. And the video here captures the first few days. You can see that the loosely connected human pluripotent stem cells, within the first 24, 48 hours, they start to open small cavity. It's going to replay. So open small cavity, you can see that. Soon after, the small cavities will grow, eventually will fuse. And by day 3, day 4, each tissue will contain a single continuous lumen cavity span across the entire length of the tissue. And the video, the second video here, capture basically the second step here. And we have one single tissue patterned into the channel. And again, this is the second step here. And you might notice a few things. In fact, there's rapid expansion of the rostral region. This is the brain-like region in the tissue. And second, there's also continuous elongation as the caudal end. Yep, Maha.
Fusion, is it polarized, meaning it starts at one location rostrally and then moves caudal, or vice versa, or is it random?
No, I wouldn't say. Well, first, it's not random. And second, also, well, let me describe. Along the entire length of the structure, and we start to see lumen formation spontaneously emerge. And it also turns out the location, the position, the location where individual lumens start to nuclearize and develop, it appears to be dependent on the confinement, the width of the tissue. And we see regular patterns of the lumens.
The cells are typically basically polarized relative to the lumen?
Yes.
Does it start out being polarized, or does it spontaneously polarize before lumen formation arises?
That I guess I don't have data to specifically answer that question, but we know that once lumens start to emerge, even as an early stage, the apical basal polarity, the apical markers start to accumulate as the lumen, individual small lumen. So that really suggesting that, I guess, apical basal polarity, epsilonization, and play a role for lumen formation, initial lumen formation. Yeah. Right. Okay. But this is not completely surprising, and because even from the previous videos, we know somehow human pluripotent stem cells, they are epsilon cells, basically.
And the rostral caudal patterning you said before, I forgot, sorry, you use retinoic acid or FGF or both?
For rostral caudal patterning, we're using Wing FGFA retinoic acid. And again, you can also argue, and this is a very simple picture, it's only 3 morphogen signals, but it works. And does that mean that we are really recapturing the local in vivo complexity? Hardly so. I don't think so.
Critical gradient that you need in order to actually get that patterning?
Definitely the patterning. I will show some. I will just show the results here. Oh, I don't have that results. I think I removed some. For sure, I think the patterning of the forebrain, midbrain, hindbrain, and spinal cord regions based on the gene expression patterns, critically dependent on the morphogen signals. You can further enlarge increase the morphogen signals to make it steeper, and that definitely will have drastic impact on the local gene expression patterns. Yeah. So it's morphogen signal dependent.
But is there a critical amount of gradient, critical gradient that you need in order for that patterning essentially?
I think the answer is yes. Yeah, the answer is yes. Yeah. I removed certain data, and we also, I think I should add it back. We also see very, very, very interesting, if you're ever interested in Hox gene, and in our system, I believe that our system is a good system to study Hox gene development. And Hox gene, we see it's amazing Hox gene collinear development of Hox genes in our system. And yeah. All right. So going back to, let me go back to the video. I'm going to replay. So it's going to replay. Yep. So that's rostral expansion. That's the brain-like region. That's called the elongation. In fact, this is the tail butt. This is the tail butt of the spinal cord. And the other feature, in fact, if you watch very, very carefully, it turns out we have delaminated neural crest cells across the entire neural axis. across the entire neural axis. And neural crest cells, and even following the previous discussion, these are very, very important cell type to give rise to a lot of connective tissues in the head, for example.
Because, you know, there is this kind of debate on the activation transformation model and whether these are two separate compartments and the forebrain and the rest.
Unfortunately, I don't think our model can answer that provide concrete.
If the anterior cells are the same as the one in the forebrain of the embryo, that suggests that with the same population, we're able to give rise to both.
Yeah. As it is in our hand. But I have to be careful though, because unfortunately, again, I removed all the Hox gene data. It turns out in our system, the most caudal Hox gene we can get is HOX9. Unfortunately, somehow in the field, the whole field has a tremendous difficulty to get cells expressing even more caudal Hox genes. I would be much more confident to answer your question if we have much like caudal genes expressed.
But it's more on the anterior side. The fact, do you have a limit, like, you know, the one genes, where do they stop? The parallel ones, anteriorly, they don't go to the tip. I suppose you have a Hox. I think the important fact is that you have a significant Hox-free region, which would be this forebrain.
That's right. Yes, yeah. So nonetheless, I guess for Olivia's question, I guess indeed, I think our model, as it is our model suggesting that the same cell population, homogeneous cell population, they have the potential to give rise to both brain cells and spinal cord, the most rostral spinal cord region, which is, I think, our data suggesting that.
What I find really spectacular here, it's the same was true for the SOMITE study, is that there's no time information. It's purely spatial, because all your cells, you see them, they're at the same age, and you can pattern them this way, exactly the way the embryo does it by playing time. So that's...
Yeah. All right. Then going back to, of course, then we can perform more detailed characterization by performing sectional images, like section the tissues, then perform conventional immunostaining. And this is the forebrain region, and you can see very nice pattern gene expression, PAC6, followed by DLX2, NKX2.1, and again, mark the subpattern region in the forebrain. In the spinal cord region, and this is I guess, a panel of genes you might be more familiar with, and you can see that PAC3, OLIG2, and followed by NKX2.2 and FOXA2. Very nice pattern gene expression. I should point also out SOX10 neural crest cells, and they're delaminating from the dorsal pole. So I will come back to the neural crest because it turns out it's a very interesting cell type, and it's interesting biology as well we can study, and also modeling disease.
I don't remember if it's in the paper, but if you wait long enough, do you have post-mitotic neurons? And like, for instance, you have a floor plate, do you have like commissural neurons, or they don't make it like...
We have not get commissural neurons, but indeed the prolonged culture, so far in the paper, we demonstrate that we culture them up to around 40 days. And we have single-cell sequencing data. Let me go to the single-cell sequencing data. It's towards the end. I think the supporting figures. Yep, this is the data. So even in our day 21 data, you can see this is the single-cell sequencing data, and this is from our model, and this is a comparison with the human reference. And indeed, you can see, just focus on this data here. And we do have, for example, emergence of secondary signaling centers, floor plate, and also, for example, asthmic organizer, they will emerge early, but then later time points, and they don't show up in the model. But we have not, and we do have post-mitotic neurons, and as around, I would say, day 40, but we have not, it's ongoing effort. We want to prolong the culture longer and longer to see whether, again, as I mentioned, that since we have different compartments, different regions, and whether we can really model interregional interactions. I think that will make this model even more powerful. And I think and when you talk to developmental biologists, you say early patterning, they say, okay, that's cool. But they don't really, I guess a lot of them would be more excited if you really show functional features associated with different organ development. Yeah. All right. And you can see this is the roof plate, floor plate neural crest, and postmitotic neurons. And as I mentioned, asthmic organizer as a secondary signaling center. And NMP population, in fact, I should point out, we have NMP population. I will go back to it. And this become there are a few very interesting cell types that we can start to zoom in to understand, to really leverage our model to study them. And that's the beauty of such models. So it turns out we decided to trace the two populations we decided to trace as to show, I will show data. One is called NMP, neuromesenchymal progenitors. I'm sure you heard of this population. It's very important and it's bipotent and will give rise to the cells in the spinal cord and also give rise to the cells associated with the somat, for example. And it has unique markers. And for example, they will co-express Brachyuris and the SOX2. And indeed, when we stain for these markers, we see a unique population as the caudal end of our model. And to check the development, the lineage development of this unique population, and again, we use the Cre line, and in this case, it's driven by TBXT. TBXT is the gene name for Brachyury. And the NMP population in the entire neural tube is the only cell population will ever express Brachyury. So this really allows us to check the progenies of this population. And you can see it works out, this reporter line works out really well in our system. So you can see the progenies of this population will continuously be generated as the caudal end, then participate in the elongation of the caudal end. And more importantly,
but it should label your entire neural tube.
No, not entire neural tube.
That means that the rest of your spinal cord doesn't derive from NMPs.
We suspect that's the case. Yes. And also point out, in fact, that I kind of neglected some details. I don't think our culture environment is most conducive to promote the long-term maintenance of this unique cell population. Because when we stain the tissues for this lineage markers, and I would say this is the data from day 4, but by day 7 or day 14, this unique population will disappear. So that really suggests that our culture system, the protocol we have, is not conducive to promote the maintenance of this population. But nonetheless, we already start to see, I hope the video gonna. Yeah, this is what I'm gonna show next. Yes, thank you. Yep, indeed, that's something. So let's watch the second video here. In fact, it's gonna play. It turns out, indeed, we believe that from our live imaging data, the lineage tracing, that really suggests that, let's watch very carefully the green cells. And this is the dashed line mark the boundary of the dorsal spinal cord. You can see that the progenies of the NMP population will give rise to the neural crest cells. And this is something very interesting. And we do, of course, remain unvalidated. We have not validated in vivo, but given our data, we suspect that and the NMP population will give rise to chunk neural crest cells, And the next question is, as I mentioned, that neural crest has very interesting biology, very interesting biology. It turns out there's axial position dependent neural crest biology. And the neural crest cells in the brain region, they will be biased towards mesenchyme fate. And that's known from animal studies, where the neural crest cells in the trunk region, the spinal cord region, they are more biased towards neural phase. And we still don't understand why, likely involving, I guess, the spatially patterned gene regulatory network. But this is the schematic, and basically, this is the existing knowledge that recently been generated. For example, again, the cranial neural crest cells, they are biased towards mesenchyme. They will give rise to, for example, TRIS1 positive mesenchyme, while the chunk neural crest cells, they don't do that. So indeed, we decided to look into whether our model will recapitulate this interesting axial position dependent biology or not. Indeed, only in the rostral region we see TRIS1 positive neural crest lineage. Well, only in the caudal region we see FOX2B positive sympathetic neurons derived from the neural crest lineage. Well, ISO1 SOX100B, these are associated with Schwann cells and also sensory neurons, and we see these cells across the entire neural axis. That really suggesting that our model might be useful to study the axial position dependent neural crest development, and we're working on that right now. It's not easy, but I think there's some opportunities there. Just to, okay, I think this might be my last slide. And now, in fact, the neural crest, there are a lot of disease modeling opportunities and using our model because there are a lot of disease associated with neuroquest development. And one of the disease, it turns out, is called CHARGE syndrome. And this is through close collaboration with my Donna Martin at UM. CHARGE syndrome, and the patients would exhibit a lot of tissue defects in the craniofacial region, and many of these tissues are associated with neuroquest lineage. So that really suggesting that CHARGE syndrome is resulted from neuroquest defects, neuroquest developmental defects. In terms of CHARGE syndrome, it's mediated by one mutation, gene mutation, CHD7. It's a chromatin modifier, and which means that it doesn't have a specific function. It's a chromatin modifier. But nonetheless, we can obtain iPS cell lines from CHARGE patient, and we can generate isogenic control. And so you can see that in the cranial region marked by OTX2, the SOX10 positive cells, this is unpublished data, and the SOX10 positive neuroquest cells, they show very different, after specification, they show very different behaviors. Only the isogenic control, they start to delaminate and they were spread out. Well, the CHARGE iPS cell-derived neuroquest cells appear to, they have difficulty to undergo EMT, delaminate, and undergo continuous development. And further, prolonged culture, in fact, in our system, and we see drastic reduction of TWIST1 positive mesenchyme and ISO1 positive sensory neurons in the model. And this has been recapitulated in our mouse model for the CHARGE syndrome. And we have the knockout, and this is the control, and you can see very consistent results. So I think with that, I would like to, I'm not sure, I will just skip this one, maybe just for fun. It's just for fun. I want to get some feedback from developmental biologists in particular. Oftentimes, they don't like me to talk about this. But nonetheless, we understand that from classic developmental biology studies, developmental biologists have developed many, many different model systems. I think this is a very, very fair statement for many problems, and there's animal on which they can be most conveniently studied. And this is what we call principle. And a very, very, I think, very accurate statement, right? We have many, many different model systems. But I would say that now, given the emergence of stem cell-based models, I will argue that, and hopefully you will agree, for certain problems, stem cell-based models may be available on which they can be conveniently studied. And I hope this is not too offensive and for my developmental biologist colleagues here. And of course, there's still a long way to go, and I think there's a long way to go, particularly as a community. I think as a community and working in this field, there's a lot of room for growth, but there's also a lot of responsibilities. All right, I'll stop, and this is my group. I'll just stop here. Yeah. Thank you.